Welcome.
Thank you.
Good to have you.
Thank you.
Well, you sequenced microbiome DNA at scale. What's the purpose here? Talk me through it.
So we did start sequencing the microbes, the bacteria from the intestinal content from farm animals, so especially chickens. It was about like 7-8 years ago, and technologies actually of sequencing really started to take off more than 10 years, and now it's very accessible, and also it became much cheaper to sequence. So that's what we do. We have now two platforms, sequencing platforms, one in Austria, one in France, and we collect intestinal content, excreta from the chickens from our customers, and we do sequencing so we could identify the microbes in the gut and try to address some of the problems that the customer may face.
Okay. Having this large library, what type of possibilities does this open up?
So that was one of the challenges when we started this microbiome customer service 6 years ago, because we had almost no samples from customer conditions. So over time, building this database, collecting more samples, allowed us to be more confident on the results, to do benchmarking with poultry industry. So I can really see the beginning as being a bit insecure when talking to customers about the data, because we had maybe a few hundreds samples in our database coming from different regions, different type of production, different ages. So the microbiome is very dynamic. And now we have like more than 6,000 samples in our database, and that's really what makes us stronger in terms of talking about sequencing, about microbiome data, and the findings that we do on those data sets.
Mm-hmm. And I think in context with that, you developed an internal reference database, correct?
So you mean a chicken or database in the gut?
That's a good question, because I only saw the term internal reference database.
No, I can explain maybe a bit more.
Yeah, okay.
So earlier I was talking about that database, a customer, more like a customer sample database, so really come from chickens. But we also developed, when we talk about sequencing, so you need bioinformatic pipelines, bioinformatic tools, and when you sequence, so you get those, there's really a lot of sequences, and you have to identify basically to which that sequence belong to. So for instance, some microbes or Escherichia coli or something like this, you need to map them to a database and to a DNA database. And we have developed also our own database. So what we do, we assemble genomes from microbes all together, because public database, they are not that complete, especially for farm animals. So we did our own internal database. So now when we have the sequences from those chickens, we can increase the rate of identification in terms of bacterial species in those samples.
Mm-hmm. Okay. And standardization here is a word that's mentioned quite a lot around databases and collection of data.
Yeah.
What makes it so important?
I think since we really moved to big data since more than 5 years, it's very important to standardize. So basically to have the same format structure of your data. If you want to have a database for broiler chickens, for microbiome in broiler chickens, it's important to have the same type of data. You cannot pull data coming from one study using a certain methodologies, then another study with maybe a different approach of sequencing, and then pull all data together. That won't work. So we always use the same workflow. We apply the exact same step on a sample, the same for also the data analysis, so we can pull all the data together. And for me, this is one of the key. This is not only about collecting data and having the biggest database. This is really to have the good quality of data and data in the same format, so you can really extract some value from those data.
Yeah. You work in precision services, right? So the entire idea of this work is obviously to add another toolkit to give us more depth, to give us more granularity. Um, how do these microbiome results integrate with other data sets like performance data sets or veterinary records?
I think that's this is also what we realized, that just microbiome data alone is basically a bit useless if you want if you want to extract a value from those data. Um, you may have data and you see it going up or down for some bacterial populations, but if you don't integrate or connect with other type of data, experimental data, observations from the chickens, phenotype as we call it, animal health, also over like animal health observation or blood markers as we do, it's very difficult to make a solid story and to really be able to address maybe customers' issues if you have just that data set alone. So that's why also in our database, we always now collect microbiome data, blood markers data, and animal health observations. And by basically associating all the data together, we are able to address specific points or to better understand some of the issues that the customer face in the production.
In that context, you shared a case study with us, lactic acid bacteria as key indicators, but basically the idea was to illustrate the approach, to illustrate the data capabilities. Talk us a little bit about through the case study and what your intentions were there.
Yeah, that was a very interesting case study because, as you mentioned, lactic acid bacteria, they are known as beneficial bacteria. You do have lactobacillus for probiotics in your yogurt, and in that study, we realized that the proportion of those lactic acid bacteria they were quite high compared to other trials, other customer trials. So we have, as mentioned earlier, we can compare basically the proportion of those bacteria to all our customer trials from different regions at different ages. And what we saw when comparing with this database, they were very high. And the thing is, the data from the same individuals, so from the same chickens, in terms of blood markers, also showed acidosis. So this is basically a lower pH in the blood. And by doing data exploration, so the team behind for doing data exploration and connecting all these associations, they also suggested that when that the lactate is actually produced by the lactic acid bacteria by degrading starch, undigested starch. And this lactate, D-lactate, which is coming from the microbes, when it's too high and when there is an excess, it could go into the blood and could result into acidosis. And we see the exact same pattern actually when we have, for some farm with really high proportion of lactic acid bacteria, they had also the lowest, for instance, pH and acidosis. So the idea is really to was to okay, yeah, I maybe just
You can start again. The idea was to.
okay. So by integrating both data sets, the lactic acid bacteria and the blood data, we suggested that wait, I need to to make a
Take your time.
yeah, this is the last part of my talk that I still need to go a bit through. Yeah.
Can be broad strokes, can be in your own terms.
Yeah.
Just
Okay. So by integrating the data from lactic acid bacteria with the blood data, that gives you a different picture because the microbiome data alone, you could see it as beneficial for the gut. But considering this, that when the D-lactate is really in excess, that gives you a different picture because we add this basically observation in the blood of those chickens. So what we suggested, of course, you still need validation. It's just not like, okay, we found the problem and we're going to fix the problem. But based on that, you can suggest different strategies. One of them is, for instance, to really avoid the excess of dilactate. And since dilactate is coming from starch and undigested starch that is then degraded and fermented by the bacteria, is to maybe try to break down starch earlier in the gut by adding maybe some enzyme like amylase, or to change the particle size for the gizzard that can probably like break down the starch better, especially some of the starch are more resistant. So basically, when starch reach the CICA, the intestinal content, you will have less lactate produced by the lactic acid bacteria, and then less translocation into the bloodstream.
Okay. Does feed formulation play a role here? Like particle size, enzyme inclusion, vitamins?
Yes, so this is not my expertise, but that's why actually we have really a team of nutritionists, vets also working all together on those data. So we have really the microbiome and the nutritional teams sitting all together to understand and connect. And that's what we were suggesting. So particle size with the gizzard to really break down a bit more like all these starch, adding some amylase, adding some, if you cannot like reduce the lactate, you can maybe also prevent the conversion of lactase, or you can try to convert lactate into pyruvate, which is for energy. So you can add some minerals like magnesium, some vitamins that can probably help to convert the dilactate into pyruvate, and to avoid having dilactate basically in the blood and reducing the pH, and having chickens with acidosis.
Mm-hmm. So, in a nutshell, we are basically trying to shape the microbiome metabolism, to shape the metabolism of the animal, to in the end shape our metabolism. Could you say that?
Uh, you mean when we consume chickens, or?
Yes, that it, that these positive properties that, we first instill on the level of um, healthy in balanced microbiota translate to the chicken, which in turn does translate to the end product.
Yes, indeed, to the end product. Our focus is really on animal health and performance. So to to try to understand why some of the customers have repetitive issues, and if we can really pinpoint at those issues, and if we can validate those findings again together, by trying to apply some of our suggestions, and then we follow up with the customer. We do new samplings, within the next flocks, and try to indeed get like a better end products, and also more sustainable products.
Mm-hmm. So, how do you see microbiome analytics influencing the next generation then of precision nutrition?
Mm, I think it's becoming a reality.
Mm-hmm.
I think I had a different like opinion maybe 6 years ago, and I think also in human research, this is becoming a reality. It's still very complex, and to me again, this is not only about like having the biggest or the largest database with a massive amount of data. This is really to reassemble all those data together, make sense of those data, connect with other type of data. And I think if we keep investing in our precision services as we did over the last 6 years, and standardizing and really integrating with our data, then we can become better at identifying problems, the cause, but also to suggest proper like solutions. And to me, this is very exciting. This is very complicated, but I've seen the progress in the last 6, 7 years in that area, and there's so much potential. I mean, now we're focusing on function, on who is there, but you have so much more. You have everything related to antimicrobial resistance, virulence factors. You have like you can drive probably nutrition, but you need validation. You need to validate experimentally, so it can be in our labs, in our facilities, but also together with customers, based on the feedback that we give, and the feedback that they also gave us back. So yes, I think we are quite still far of being able to really solve all the problems with those technologies, but definitely we get much better in terms of precision and much higher resolution on what is really happening in the gut or in within the animal.
Mm. Data is the key word here, I gather. And you often say, from what I could see from your presentation, that data without context is equals zero. You know, that
Mhm.
there's no value in it, which is obvious.
Mhm.
But how do we get context?
We get context by when you want to start a precision service, you need to make sure that you're not going to just collect biological data, biological samples, but all those experimental data, those observations from the chickens, from the flocks, the feedback from customers. And like I said, we to connect with other type of data. So we have those blood markers, also data that are coming from the same animals that we analyze the microbiome. And I could also give an example that I've been into research since like last 20 years. I also did academic before moving to industry, and now you need to write papers, you need to write publications, and very often you see effects. You know, you see maybe some markers going up, going down, and if you don't really collect or if you don't see really effects also on the performance of those animals or other like type of data, then you're just gonna conclude about what you see. And when you look for it, you will find something. You will find a reason why it's going up, why it's going down, maybe in another study. So that's why for me, it's what we're doing is very very important and needed because we connect really microbiome data with blood marker data, animal health observations all together to try to have a bigger picture and to make a story out of it. It's a bit like a puzzle. If you have only a few pieces, uh, it's very hard to complete your puzzle. But if you find the missing pieces basically, then that puzzle, it's you can complete it. So it's really trying to put pieces of a puzzle together, and this is a really big puzzle in our field.
So, talking about puzzle pieces, at the end of the day, we're building this puzzle for the people who work with the animals, you know, and for those who develop the feed for the animals. How do we turn how do we turn microbiome sequencing into value for producers?
I think you need to build credibility when you report to customers your findings. Again, this is an exchange. Very often, they open up to. If they see that some of your findings make sense, they will start opening up, having a discussion, exchanging more information, and this is how you can really turn this into value. So with that context, um, but this is a lot of data exploration work. You can sequencing is easy. Sequencing, it's a technology. It went very fast in terms of development, but the use of data is not at the same pace. And you get like millions and millions of data points on microbial genes, on microbes, also those blood markers. And the objective is really to connect all the points together. And once you can do that, and then once you also discuss with customers, and again, that customer maybe tells you, okay, I tried what you suggested and it seems to work, then I think this is where you can really validate and where you really build credibility on your data. But that's a very very challenging. It's very challenging, and I think this is not only in animal production. This is also in human research. Sequencing is everywhere in human research for clinical studies, but it's a challenge to really connect all the data together, and then again to validate those findings and to generalize those findings. Because maybe sometimes what you see for a customer, what can be true, is maybe not true for another customer. But also sometimes when you see some patterns, some profiles, we need also to make to try to explore if we have seen that in our big database, and if we are also able to reproduce those results.
Okay, great. Considering the technological advancements on the level of data processing, data collection, AI in the last 5 years, what excites you most about this technology at this certain point?
I think this is we are using the most recent technology of sequencing. So this is based on it's nanopore based, and I will show actually tomorrow at my presentation. We have this very small portable sequencer, and basically anyone can sequence and anytime anywhere, and that makes things like for me always when I process my samples, when I'm ready to put my samples on this sequencer, I still have the same feeling that sequencing is very accessible, and it's such a good feeling that you can sequence within a few days. I can sequence basically your genome within 2 days. I can do that. I can then process those data. We have also the chance, I think, in our company to have a very strong expertise in terms of bioinformatics, in terms of building infrastructure. So infrastructure behind data, but also pipelines, tools. And they made it accessible to everybody because I don't have a background into bioinformatics, but with just some basics, I can process my data and I can really analyze and report those data by myself, which is what is very exciting, on top of being super fast.
Yeah. So we seem to be at a point where everything starts to come together.
Yeah, yeah, I think so. I think it's it's really a world of big data. It has pros and cons. I think I will touch a bit that in my presentation tomorrow. AI can be scary, but AI can provide or not only AI but a supercomputer. So we also it's really incredible the achievement we've done with a supercomputer with chips that are basically behind all this AI and algorithm. Everything is so much faster, but everything is so much easier for us scientists today that it's I see it as positive for now actually.
Sounds good. May it get ever easier for you. Bertrand, thanks very much for your time.
Thank you. Thanks for the
And I'm looking
invitation.
forward to your presentation.
Sure.